Already we have discussed about how pollen pistil interaction takes place and how the pollen grain is carried from anther to stigma. Now it's time to discuss about double fertilization. Double fertilization, a unique type of fertilization which is taking place in angiosperms. Already you know this structure. It's a carpel having the flat form to receive the pollen grain, that is stigma, and a long style and an ovary. In this ovary there is an ovule with embryo sac, that is a female gametophyte, in which towards the micropylar end group of 3 cells named egg apparatus, in which 2 synergids and 1 egg cell, that is female gamete, and 2 polar nuclei are there. In central cell they are haploid, 2 polar nuclei, and 3 antipodals, and this embryo sac is there in nucellus. Already we know in 60% of angiosperms pollen grain is transferred and fall on the stigma in the 2-celled stage. Those are vegetative cell and generative cell. In 40% of angiosperms, a pollen grain falls on the stigma is 3-celled, where that generative cell gives 2 haploid male gametes, and that vegetative cell makes a long pollen tube. That pollen tube passes through the stigma and a style, enters into ovary, and through micropylar end it enters into embryo sac. Already I have told, in most of the angiosperms the entry of a pollen tube into the ovule through the micropylar end, that is said to be porogamy. Rarely this pollen tube can enter into the ovule from chalazal end only, that is said to be chalazogamy. In other cases this pollen tube may enter in through the integuments, then it is said to be mesogamy. But more commonly porogamy takes place. It means the pollen tube, which is carrying 2 haploid male gametes, enters into the embryo sac through the micropylar end. This pollen tube first enters into one of the synergids, where in synergid filiform apparatus is there that guides pollen tube to enter into synergid, so that these 2 male gametes are released in embryo sac. Already we know one female gamete, that is haploid egg cell, and 2 haploid polar nuclei, and 2 male gametes enter into embryo sac. Let us explain by drawing this embryo sac separately. This is embryo sac. Here 2 synergids, they start degenerating, and this is egg cell, haploid egg cell it is a female gamete. And these two nuclei, these two male gametes, now they have entered into embryo sac. One is here. This is one of the male gametes, haploid, and another male gamete is here. Haploid, and two polar nuclei in central cell. These are also haploid. And three antipodals start degenerating. Here, this haploid male gamete fuses with two haploid polar nuclei. Two polar nuclei, leading to the formation of a triploid nucleus. That triploid nucleus is primary endosperm nucleus. As three haploid nuclei get fused, hence it is said to be triple fusion. It is said to be triple fusion. And this male gamete fuses with egg cell, leading to the formation of diploid zygote. That is said to be syngamy. Syngamy. That is said to be syngamy. And one more interesting thing is, two separate fusions are taking place here. One is syngamy, that is, haploid male gamete fuses with haploid egg cell. That is syngamy. And another fusion where three haploid nuclei get fused. Hence, it is said to be double fertilization. Double fertilization. Fertilization in angiosperms is triple fusion and double fertilization. They may ask two marks question. A fertilization in angiosperm is said to be triple fusion and double fertilization. Why? It is said to be triple fusion because three haploid nuclei get fused. Those are one male gamete and two polar nuclei. That is triple fusion, leading to the formation of a triploid primary endosperm nucleus. Primary endosperm nucleus. Then, why it is said to be double fertilization? Here, two fusions took place. One is the fusion between male gamete and egg cell to form diploid zygote. That is said to be syngamy. And another fusion, a fusion of three haploid nuclei. As two separate fusions are taking place, it is said to be double fertilization as well. Whenever the question is asked, we must explain in this way. Here I have drawn the nuclei which are about to fuse, and the same embryo sac I am going to draw here, where we would see the products of their fusion. Here, three antipodals get degenerated even two synergids get degenerated, and this male haploid male gamete fuses with haploid egg cell, leading to the formation of zygote. This is zygote. It is diploid, and one haploid male gamete fuses with two polar nuclei, leading to the formation of a triploid nucleus. This triploid nucleus is named as primary endosperm nucleus, that is PEN. It is a triploid nucleus. It is a triploid nucleus, and this triploid nucleus is there in central cell. Now this central cell is said to be primary endosperm cell, that is PEC. It is an example for triploid cell. Example for triploid cell. With these two diagrams, there is a clear cut explanation about triple fusion and double fertilization in angiosperms. Why it is said to be triple fusion? One male gamete, haploid male gamete, fuses with two polar nuclei. That is said to be triple fusion. Due to this, the formation of a primary endosperm nucleus named PEN, it is triploid. 3N, 3 sets of chromosomes. And why it is said to be double fertilization? Here two separate fusions are taking place. One is the fusion between male gametes and egg cell to form zygote. One. And then the fusion between three haploid nuclei is another fusion. Hence the name double fertilization. So that in this embryo sac, a female gametophyte, after fertilization, these three antipodals get degenerated, two synergids get degenerated, and a male gamete and a egg cell fuse to form a zygote, which is named as syngamy, and one male gamete fuses with two polar nuclei, forming primary endosperm nucleus, is a triploid nucleus, and this central cell becomes primary endosperm cell, PEC, that is a triploid cell. And they may ask the question, which of the following is a triploid nucleus? That is primary endosperm nucleus, triploid nucleus. And the next question, which of the following is a triploid cell, that is primary endosperm cell, is a triploid cell. And remember, from this primary endosperm cell, there is a formation of a triploid tissue, that is endosperm. From this, after fertilization, endosperm will form, and is an example for triploid tissue. The example for triploid nucleus, primary endosperm nucleus. The example for triploid cell, primary endosperm cell. And the example for triploid tissue, that is endosperm, is a nourishing tissue. It provides nourishment during the development of embryo from the zygote. And let us discuss about a post-fertilization events. Post-fertilization events. It means events after fertilization. 1. Endosperm development. Second, embryo development. Formation of seed from ovule and formation of fruit from ovary. These are the post-fertilization events. Endosperm development, embryo development, and a formation of seed from ovule. After fertilization, the whole ovule becomes a seed, and a formation of fruit from ovary. That is, after fertilization, ovary becomes fruit. First, let us discuss about endosperm development. They may ask, what are the post-fertilization events? Endosperm development, embryo development, formation of seed from ovule, and a formation of fruit from ovary, collectively named as post-fertilization events. These four together collectively named as post-fertilization events. It's time to discuss about endosperm development. Already we know that when one male gamete and two polar nuclei fuse to form PEN, it must be triploid, primary endosperm nucleus, and this is there in the cell. This cell is PEC, is a triploid cell, that is primary endosperm cell. From this only we do expect the formation of endosperm. Formation of endosperm. We do expect the formation of endosperm. It's a triploid tissue from this. Endosperm is formed from primary endosperm cell. It is having primary endosperm nucleus. This primary endosperm nucleus is formed during triple fusion, that is the fusion between haploid male gamete with two haploid polar nuclei. And how this endosperm, already we know endosperm is a nourishing tissue. It provides food, nourishment to the growing embryo from zygote. And how this endosperm is formed. Let us see here. This is PEC, a primary endosperm cell. it is having that triploid nucleus, that is PEN. How this endosperm is formed? First, this primary endosperm nucleus undergoes a free nuclear division, so that totally in this PEC, triploid free nuclei are formed like this. Free triploid free nuclei, that is due to free nuclear division. Gradually, what happens is a cell wall will be formed around these free nuclei like this. Here, totally free nuclei are there, and this you can say free nuclear endosperm. And a cell wall formation starts around all these free nuclei. A cell wall formation starts, leading to the formation of cellular endosperm. Cellular endosperm. It may take place continuously and totally becomes cellular endosperm. At the starting, it is a free nuclear endosperm. Even only free nuclear endosperm will be there, or a totally the cellular endosperm due to the formation of cell wall around free nuclei. Whether it is free nuclear endosperm or cellular endosperm, both of them are triploid tissues. Both of them are triploid tissues. And you might have noticed in coconut, both free nuclear endosperm and cellular endosperm. In coconut, we can see coconut water, but coconut water is nothing but free nuclear endosperm. It is having a free nuclear of triploid nuclei, that is free nuclear endosperm, and there is a tender white kernel, that is cellular endosperm. In that coconut, there is a white cellular endosperm. So that in case of coconut, we do find both the endosperm, free nuclear endosperm in the form of coconut water, and cellular endosperm in the form of a white, a tender coconut, which is made up of cellular endosperm as well. In this way, hence the formation of endosperm. And it may continuously by cell wall formation that free nuclear endosperm becomes cellular endosperm. This becomes endosperm as well. It is triploid tissue, is a nourishing tissue. It is filled with reserved food. That food can be provided to growing embryo. That food can be provided to growing embryo as well. How that endosperm formation takes place? See here, 3 haploid nuclei are fused during triple fusion, leading to the formation of primary endosperm nucleus, which is triploid, and this primary endosperm nucleus becomes primary endosperm cell, and from this endosperm is formed. How? That primary endosperm nucleus undergoes repeated mitotic division, leading to the formation of free nuclei. All of them are triploid. And this kind of endosperm is named as free nuclear endosperm. It's a triploid. And gradually cell wall formation starts around these free nuclei from the periphery, leading to the formation of cellular endosperm. Total endosperm may be like this, and total endosperm with cells, that is cellular endosperm. But in case of coconut, we can notice both the endosperms. Free nuclear endosperm in the form of coconut water, and a cellular endosperm, that is a white colored kernel in that coconut, that is cellular endosperm. Whether it is free nuclear endosperm or cellular endosperm, that is the triploid tissue only, and it is filled with reserved food, and that helps for, it provides nourishment to the growing embryo. The second event, that is embryo development. Already zygote is formed by the fusion between haploid male gamete with haploid female gamete, that is egg cell, during the process singly. And once the zygote is formed, its development into embryo starts only when the endosperm gets developed. Because endosperm is a triploid tissue, is a nourishing tissue. It provides food to the growing embryo. Until unless endosperm formation starts, there will be no embryogeny. Means embryo development won't be taking place. Once the endosperm develops partially, so that zygote starts developing into embryo. And starting, that zygote undergoes repeated mitotic division, leading to the formation of a ball of cells. And once a ball of cells are formed, that embryo looks like globose, that is globular embryo, and this suspensor pushes that embryo deep into the endosperm to get the food for growing cells, for multiplication. And this globular embryo gradually becomes heart-shaped, and finally it gives rise to a mature embryo, and it may be a monocotyledonous embryo or dicotyledonous embryo. Starting stages are one and the same. That is, a single-celled zygote undergoes repeated mitotic division leading to the formation of a ball of cells. Then that leads to a globular embryo, and that is pushed into the endosperm by the suspensor, and it becomes heart-shaped, and finally embryonic axis will form, and with the cotyledons, with two cotyledons. And here we can notice a plumule part in embryonic axis. This plumule part is giving rise to a shoot system. A shoot system is formed from this plumule part of embryo, and from radical part root system is developed. And this is matured embryo, but we have got to discuss about separately the embryo of monocots and the embryo of dicots, because in later stages their development and even structure is varied. But at starting, a zygote gives rise to ball of cells by repeated mitotic division, and embryo becomes globular, then heart-shaped. Finally, the appearance of radical part which gives rise to root system, plumule part which gives rise to shoot system, and with the cotyledons. And we have to study separately a structure of dicot embryo and structure of monocot embryo.